Hello and welcome to another exciting edition of Biology Podcast with Mr. Zabel. In today's lesson, what we're going to be looking at is human genetics. Now, we've talked a lot about Gregor Mendel and his experiments he conducted on plants and peas and flowers, but what about human characteristics? And so what we're going to be looking at in today's lesson is specifically looking at human genetics. So one thing you have to keep in mind is that when we look at Gregor Mendel's work, we're going to see that it's incomplete. And again, you also have to keep in mind that right now you know more information than what Gregor Mendel knew. Gregor Mendel never knew about DNA and he never knew about cells, so he really didn't truly understand where our genetic information came from. So again, although Gregor Mendel was considered the father of genetics, his work was considered incomplete because he failed to ask the big question: where specifically in the cells are these factors that control heredity, which we now know is our DNA inside of our nucleus. So with our growing understanding of DNA and the genetics behind everything, we can start to get a better understanding of what makes us human. And so one of the things that we have done as a human race is we have actually mapped out our human genome. And so a genome is basically a full set of genetic information that an organism can carry. And so in this genome, we can actually look at your specific chromosomes, and you can actually look at where specific genes reside on those chromosomes. So specifically what your hair color is going to be, where that sits on the specific chromosomes, the color of your eyes, the length of your hair, the type of your hair, all of those are going to be mapped out in what we call the human genome. And so they developed this Human Genome Project, which basically mapped all of your chromosomes out. And so when scientists look at the human genome, they begin by looking at your chromosomes. And again, we'll talk a little bit more about how we actually look at your chromosomes in order to make up our human genome. So in 1990, scientists completed the Human Genome Project. And if you take a look here, this is basically what they did in this Human Genome Project, is they basically took each chromosome, chromosome 1, all the way up to chromosome number 22, and then also our X and Y sex chromosomes. And they basically just mapped everything out. And they looked specifically at each chromosome and looked at specifically where these specific genes sat on the chromosome. So where specifically is eye color, where specifically is hair color, where specifically is the type of hair that you're going to have. So if you look at the link below, if you're on Sophia, you can also look at a YouTube video that will basically talk more in depth about how they actually conducted the Human Genome Project and how they actually mapped out each and every single chromosome on the human genome. So how do scientists go about mapping our human genome? Well, one of the first things they have to do is they have to create what we call a karyotype. And now we've looked at karyotypes before. And if you remember, a karyotype is basically going to look at our chromosomes within our cells during mitosis. And specifically, it's going to look at the stages of metaphase, where the chromosomes are going to line up in the middle. And so we want to look at those chromosomes in the middle because they're the easiest to see at that point in time. And then scientists will basically cut those chromosomes out and they'll take a photograph of the chromosomes, and they call this picture a karyotype. And now remember, the karyotype is going to show the complete set of chromosomes, all 46 chromosomes or 23 pairs of chromosomes. And then remember, those are going to range from the largest being number 1 all the way down to number 22, which will be very small, and then it'll have our 2 sex chromosomes. So if we take a look here, we're going to see a typical karyotype image that you would see. Again, if we look at this, it's going to show all of your chromosomes, all 46 or 23 pairs. Again, you're going to have homologous chromosomes, one from mother and one from father, and they're going to be arranged from largest being number 1, and they're going to decrease in size until we get to number 22. And if we take a look at this individual, we're going to have our sex chromosomes be here. And again, these are going to be the same for this individual. And so both of these are X's, meaning that this is going to be representing a female. Again, this is what a karyotype is going to typically look like. And again, we see this during metaphase of mitosis. So when we look at a karyotype, we can see that there's going to be 2 divisions, and you can see one of the divisions are going to be here in pink, and these are called your autosomes. And again, we've mentioned this before, so it should be a review, but there's going to be 44 or 22 pairs of autosomes. So these are going to be all of the chromosomes besides our sex chromosomes. These 2 sex chromosomes over here can either be, you can have 2 X's, which would make you a female, or you could have an X and a Y, which would make you a male. So this individual would be a male. So again, those 2 chromosomes out of the 46 are going to be sex chromosomes. Again, your sex chromosomes can be X or Y. If we take a look here at your female is going to have 2 X's. So she's going to be XX, whereas the male is going to be XY. And when we talk about human genetics and how we determine the sex of a child, well, this is going to be very important. Remember, we have to pass down one of these alleles to our offspring from mother and one from father. The female will donate an X. Whether it's this X or this X, it doesn't matter. If she donates one X to the girl or the boy, she's going to only be able to donate this one X. The male, on the other hand, is going to determine the sex of the child, because the male, if he donates his Y, he's going to create a boy, and if he donates his X, he's going to create a girl. So again, it's the male that is determining the sex of our child. So like I stated earlier, Gregor Mendel was considered the father of genetics, but he failed to ask the big question: where specifically is this genetic material? And so he really didn't have a full understanding of DNA, genes, and chromosomes. It wasn't until the scientist by the name of Walter Sutton came into the picture, where he was looking to use microscopes to look at sperm formation in grasshoppers, where he came up with this theory, which is called the chromosome theory of heredity, which basically states that genes are located on chromosomes and each gene occupies a specific space on that chromosome. And so if we take a look here at our picture, again, we would have, let's say these are two chromosomes, one from mother and one from father, and we would have specific genes that would be same corresponding from one chromosome to the other. And so if we looked at this one, we could see that maybe this is for eye color, maybe this is for brown eyes, and then on the other chromosome, it's in the same location. So the color for your eyes is going to be determined by these genes that are located right here on the chromosome. And again, that's what we saw when we mapped the human genome, is that eye color would be on a specific chromosome in a specific location. So a scientist that actually looked at gene linkage was a scientist by the name of Thomas Hunt Morgan. And Thomas Hunt Morgan basically devised experiments looking at fruit flies and looked at how these genes were linked. And so he finally came up with what linked genes were. And he noticed that some genes on chromosomes are linked together, meaning that they are inherited together. That one gene, whether it is the color of the fruit fly and the wings, they are linked together, meaning that they undergo independent assortment. So in Morgan's fruit fly experiment, he first looked at two genes. The two genes that he looked at was the body color, and he also looked at the wing size. And so when he looked at the body colors, he was determining whether they were gray or black bodied, and the wings were either going to be normal or small wings. And so what he started off with his parental generation was he looked at a purebred fly that had a gray body and normal wings, and he crossed that with a black bodied small wing. When he looked at coming up with his letters, again, he has his big G, which is going to represent gray, which is the dominant allele, over black, which is represented by a little g. His normal wings were represented by a capital W, which were dominant over the small wings, which were a lowercase w. When he crossed these together, these two flies, again, he did a dihybrid cross to figure this out, because we're dealing with two different traits or characteristics that we're going to be looking at: body color and wing size. And then when he did his dihybrid cross, he ended up getting 100% of them were going to be gray and normal to wing. And again, that's exactly what he saw in his results. He then decided to take another step to get his F2 generation. And when he did this, he crossed his normal grayed body and normal wings. He crossed those flies with black small winged flies, which was represented as little g and little w. Again, he did a dihybrid cross and figured out the ratios, and he got the expected ratios of having 25% ratios for all of his flies. However, when he actually did and performed the test, what he found out was that he was getting results of 41.5% of them were gray and normal, another 41.5% of them were black and small, and then 8.5% of them were black and normal, and then another 8.5% of them were gray and small. So he did not get the desired ratios which he expected. So this basically said that these genes were linked together. So again, when Morgan conducted his experiment, he expected to see these ratios, these 25% ratios, according to the dihybrid cross that he conducted. However, when he actually performed the experiment, he did not get these desired ratios. He got the 41.5% down here, and then he got the 8.5% over here. So these results indicated that the genes for the body color and the wing size were somehow connected to one another. So he called these linked genes. And these linked genes did not go through independent assortment. They didn't go individually. Instead, they went together. So if genes are linked, why aren't they linked all the time? Well, this is explained through the process of crossing over. And we've already talked about crossing over if you remember back when we talked about meiosis. Crossing over is a process that takes place during prophase 1 of meiosis, where these two homologous chromosomes come up to one another and they exchange genetic information. As they do this, they turn into what we call recombinants, and this is called a recombinant DNA. And so as we turn into these recombinants, we genetically shuffle and basically make genetic variation in our genetic material. And this is why our genes aren't linked together. On the same lines as talking about linked genes, we also have what's called sex-linked inheritance. Because the X and the Y chromosome determine the sex, the genes located on them show a pattern of inheritance called sex linkage. And so when we look at sex linkage, we're basically looking at the genes that are located on these sex chromosomes. In humans, they're going to be on our X and Y chromosomes. And so it wasn't until Nettie Stevens noticed that when he was looking at his flies, they either had red eyes or white eyes. And when he looked at where that gene was being expressed, he noticed that it was being expressed on the sex chromosomes. And so that's where we're going to come up with this sex linkage. So Thomas Hunt Morgan devised an experiment to look at sex-linked gene expression. And in this experiment, he looked at fruit flies, and he specifically looked at their eye color to see if they were white-eyed or red-eyed fruit flies. And so if we look at eye color, it's going to be determined by our X chromosome. In males, they have a Y chromosome, but it's not going to be functioning in determining the eye color. In females, they'll have two alleles that will express eye color. And again, if we look at his parental generation, he's going to cross a homozygous red-eyed female with a white-eyed male. And so as he crosses these, he gets his F1 generation. And whenever we write this in terms of writing an experiment in sex-linked genes, we need to always write it like this, where we're going to put the sex. So if we have a female, she's going to be XX. And then we're going to represent the alleles that she'll have. So red-eyed would be big R, so she has X big R, and then white-eyed would be little r. The male, on the other hand, is going to be XY, so those will be the chromosomes that the male has, and then the male can only receive one allele that's going to express the eye color, so we have a capital R for red, and so we put that here. And again, if we look back at the parents, we can see that this female has to donate an X to the female, so it would donate one of these, and this male has only one X, so he has to donate this white one. So we get a heterozygous female down here that's going to be red-eyed. This male has to get the X from the female, and it's going to automatically be red-eyed. So we're going to see all of his flies in the F1 generation are going to be red eyes. And so he decided to take his F1 generation and cross it and see if we can get any white-eyed flies. And so what he looked at was he crossed these two together, and he ended up getting one of the males is going to be white-eyed, another male is going to be red-eyed. He's going to have both females will be red-eyed. One will be heterozygous and one will be homozygous. And again, if we take a look back at the parents, we're going to see that this male has a non-functioning Y that he has to give to both of the boys. And then this female, again, is going to either pass off her white-eyed or she's going to pass off her red-eye. And again, this is the female in this case is determining the eye color of our males. It's not the male, but the female, the one that decides it. And then again, if we take a look at our females, this male always has to donate this red eye. So all the females are going to be red-eyed no matter what. And so that's what sex linkage is, and we're going to see how we can use that to determine other effects in humans. So this understanding of sex-linked gene expression is very important because we can use this information to determine human genetic disorders. One of the type of human genetic disorders that we're going to look at is called sex-linked genetic disorders. And so when we take a look here, we can have an unaffected male that's going to have the unaffected gene expressed on his X chromosome, and then the mother is going to be a carrier. So again, on one of her X's it's going to be unaffected, but the other X, it will have a affected genetic disorder that could be passed on. And so this can be very dangerous because the male, again, is going to always pass on to his offspring the unaffected disorder. But the female, on the other hand, if she passes her affected disorder to her children, the only ones that will be affected are going to be her sons. And so we have a probability where we have a 50% chance that this disorder could be passed on from parent to offspring, and it's going to affect her sons more than it would affect her daughters. Because the daughters, again, are going to automatically receive one X from their father. And so that concludes our lesson for today. I hope this was a helpful podcast, and until next time, remember, keep studying biology.